You know, every single time you learn something new, whether it's someone's name, a new recipe, or a wild fact, your brain is physically changing. But how? I mean, how does that actually happen? Well, in this explainer, we're going to crack open the brain's learning code and see exactly how a memory is born. Just think about that for a second. An experience, it's over in a moment, right? But the memory of it can stick with you for your entire life. How is that even possible? It means something inside your head, something physical, has to be altered. Now, this isn't magic, it's pure biology, and that is the central mystery we are going to solve together today.
First up, let's talk about the memory machine itself. We're going to get right to the heart of how this whole thing works.
The entire process really boils down to this one core concept: synaptic plasticity. Now, I know it sounds super complicated, but the idea is actually pretty simple. The connections between your brain cells, your neurons, they aren't set in stone. They can get stronger or they can get weaker, all based on how much you use them. This is the biological bedrock of all learning and memory. So where does all this strengthening and weakening take place? It all goes down at the synapse. This is this microscopic gap between two neurons. You can think of it like a handshake. It's the point of communication. And for our purposes, we're going to be focusing on some of the most studied synapses in the brain, right inside the hippocampus, which, as you might know, is a major hub for memory. Okay, so if the synapse is the stage, who are the actors? Who are the key players making all the decisions? Let's meet the molecular gatekeepers that control the entire show.
On the receiving end of that synapse, there are two absolutely critical characters we need to know. First, meet the AMPA receptor. This guy's the workhorse. When the chemical messenger, glutamate, arrives, it just opens right up, nice and fast, giving a quick electrical signal. Simple. But then, there's the NMDA receptor. This one is different. This one is clever. Scientists have a great name for it. They call it a coincidence detector. And the genius of the NMDA receptor is all in its design. You see, it has this tiny magnesium ion that blocks it, kind of like a cork in a bottle. And for that cork to pop out, two things have to happen at the exact same time. First, glutamate has to show up from the sending neuron. And second, the receiving neuron has to already be buzzing with electrical activity. This is literally how the brain wires together things that happen together. It's the cellular basis for making associations. It's brilliant.
So, the NMDA receptor opens its gate, but only when two things happen at once. But what happens next? How does the neuron decide, okay, should this connection get stronger or should it get weaker? The answer, the crucial messenger that decides the fate of this connection, is calcium. As soon as that NMDA receptor gate opens, calcium ions flood into the cell. And it's the amount of that calcium that is the key to this whole mystery.
See, this is the crucial part. It's not if calcium gets in, but how much calcium gets in. If there's just a little trickle, a modest influx of calcium, say from some low-level, unimportant activity, it flips on one set of enzymes called phosphatases. And the result? The synapse gets weaker. But if there's a huge flood of calcium, caused by intense, high-frequency firing, it wakes up a totally different crew of enzymes, especially one called KMKI. And what happens then? The synapse gets much, much stronger.
These two outcomes are the absolute foundation of plasticity. Neuroscientists call the strengthening process long-term potentiation, or LTP for short. This is the cellular mechanism for making a memory. The weakening process is called long-term depression, or LTD, and that's just as important for helping us forget things or fine-tune our memories. Okay, so we've got this signal, calcium, and we know the outcome, either LTP or LTD. But how does the cell physically change the strength of that connection? How does that calcium signal get translated into real physical action? Well, it all comes back to our workhorse, the AMPA receptor. The easiest way to think about this is that the synapse basically has a volume dial, and it turns that volume up or down by physically adding or removing AMPA receptors from its surface. You know, more receptors means the synapse is more sensitive. It's like turning up the volume on your stereo. So here's exactly what happens. During LTP, that big flood of calcium is a command. It tells the cell, hey, grab those extra AMPA receptors we've got in storage and plug them into the synapse now. So now, with more receptors on the surface, the same amount of glutamate causes a much bigger response. The volume is officially cranked up. And for LTD, it's the exact opposite. That little trickle of calcium signals the other enzymes, and their job is to go out and pull AMPA receptors out of the membrane. With fewer receptors to listen for the signal, the synapse becomes way less sensitive to glutamate. The volume gets turned down. It's just such an elegant, dynamic system.
Now, turning a volume dial up or down is a functional change. But for a memory to really last a lifetime, these changes have to become permanent. So let's look at how this leads to the actual physical rewiring of your brain.
And this is where it gets truly mind-blowing. These changes in the number of receptors, they aren't just temporary fixes. They set off a chain reaction that leads to lasting, physical, structural changes in the brain. Learning isn't just a concept, it's an architectural project. You are literally rebuilding your brain with every new thing you learn. I mean, just look at this timeline. It starts in milliseconds. Neurons fire, the coincidence is detected. Within minutes, the calcium signal has already moved the AMPA receptors around, changing the synapse's volume. But then it keeps going. Over hours and even days, the cell starts making brand new proteins to make these changes permanent. The actual physical structure of the synapse can grow bigger and stronger, or it can literally shrink and fade away. Now that we understand the beautiful mechanics of it, we can really appreciate how important it is. This elegant system is the engine of learning, but it's also incredibly delicate. So let's take a quick look at what happens when it gets disrupted.
So, what are the real-world consequences when this finely tuned molecular machine for learning and memory breaks down or gets hijacked? Well, the results can be absolutely devastating.
The stakes here are just enormous. Take Alzheimer's disease. We see these toxic protein clumps that essentially jam the whole system. They block the synapse's ability to strengthen, they block LTP, and instead they push it toward weakening, toward LTD. This leads to synapse loss and ultimately cognitive decline. Or look at drug addiction. Here the system is hijacked. A drug like cocaine can actually force the insertion of new AMPA receptors, hot-wiring the brain's reward circuit and creating this incredibly powerful pathological form of learning.
And this quote from the researchers Lüscher and Malenka really just says it all. The reason scientists are so focused on this is because these mechanisms, LTP and LTD, they aren't just theories. They are very likely the actual cellular building blocks of how we learn and how we remember.
So I want to leave you with this final thought. We've seen today that learning isn't some abstract idea. It's a physical architectural process. Every memory you have, every skill you've ever learned, has physically rewired the connections in your brain. So if that's true, what does it really mean, on a deep biological level, to change your mind? Thanks for joining me.